Perfect. Hello, everyone. I just want to start by saying thank you very much for being selected to talk here today. It's a real honor to be here and talk to such a great room of people. So my name is Conor Maltby. I'm the head of translational medicine at Ulysses Neuroscience, and today I'm just going to give a very brief overview of some of the work we've been doing on the preclinical space in human biomarker analysis. Let's see. Am I pressing the right button? Just as a very quick background for those who may not be familiar with who we are, we're a small contract research organisation based in Dublin, Ireland, and we work really in the preclinical space all the way from in vitro models up to human preclinical biomarker discovery across a range of different indications from neuropsychiatry, neurodevelopmental disorders and neurodegenerative disorders with a real focus on rare diseases. So today what I'm going to go through is very quick background. I don't need to go into too much detail on, current biomarkers and clinical rating scales. We've already heard a lot from individuals here about this. Looking into α-Tubulin post-translational modifications, their context within disease, pharmacological relevance, and how they influence microtubule stability. And then two small clinical studies that we have run to try and assess whether this could be a feasible biomarker across CMT. So I'm not going to go into detail here. As I said, we have various different clinical rating scales, all with their pros and cons, and obviously a hugely beneficial tool for both clinicians and patients, and a very accessible way of measuring progression and severity. However, they do come with their limitations. They can be insensitive to slow disease progression, particularly in cases such as CMT1A. There's always a potential for inter-rater variability. But these can be strengthened, when moving towards clinical trials, when they're used in conjunction with measuring, biological biomarkers that are sensitive to, say, either severity and progression. I'm not going to go into all of the different biomarkers we have for all of the different CMTs. I'm just going to stick here with NFL as it's relevant for my talk today. So, NFL, neurofilament light chain, is a protein that associates and forms part of the cytoskeletal network, and it's very robustly released into the extracellular space of CSF and into the blood during axonal injury and during cellular injury. So it's great. We can measure this as a really robust increase across numerous different neurodegenerative disorders, but there's the limitation that it's usually quite sensitive to age. It's fairly non-specific. It captures kind of a general signature of neurodegeneration, and again is insensitive to severity or progression, as has been seen in many cases. So a quick background on microtubules. Microtubules are these hollow tubular structures that really form the fundamental network of structure and transport through the neurons. They go all the way from the cell body, around the cell body, out to the distal axon and the distal dendrite. And they really are key to maintaining structure within neurons, but also facilitating both anterograde and retrograde transport of key factors to and from the cell body and out to the areas where the stuff really goes on, the synapses and where the cells are connecting to each other. And microtubules are formed of these alpha and beta tubulin heterodimers. And these undergo various different post-translational modifications that influence how they behave. Today, I'm going to focus on this particular PTM, which is acetylation at lysine 40 at the N-terminus of alpha-tubulin. So acetylation at the N-terminus of alpha-tubulin really plays a role in how fluidic or stable microtubules are. So what we know is that when you have reduced levels of acetylated alpha-tubulin, you have reduced levels of microtubule stability. And when you have increased levels of acetylated alpha-tubulin, you have increased microtubule stability. Neither of these is an inherently negative process, and they are very important to the normal homeostatic function of microtubules and neurons. What we tend to see is that you have reduced microtubule stability during neuron growth, during neuron development, and during synaptic and neuronal remodeling. And then you would like to see, and what we hope to see is, an increased level of acetylated alpha-tubulin and increased microtubule stability in the peripheral nervous system, where you have these really long neurons projecting from the base of the spinal cord to the end of the toe, over a meter long in some cases. Now, alpha-tubulin is acetylated by this protein alpha-tubulin acetyltransferase 1, and then it's deacetylated by this protein histone deacetylase 6, HDAC6. So the reason we're looking into alpha-tubulin is, one, our background is in microtubule proteins and microtubule biology, But also, we all know over recent years there has been particular interest in this protein here. So inhibiting this protein, inhibiting the deacetylation of alpha-tubulin has been shown to be effective in various different models of CMT1A, HSPB, CMT, and also, as we heard yesterday as well, we all know, Augustine are now moving towards the clinic with their novel HDAC6 inhibitor. So, on to the first study. So this was a small pilot study conducted in a cohort of Irish patients a good few years ago, back at the end of 2021 through to the middle of 2022. And how it works with us at Ulysses is we handle all of the kind of bureaucracy, admin pieces of this. We just can't really do the sampling. We require the clinicians for that. So we, when we want to do a study with a patient foundation or a, pharmaceutical partner, we do the site selection, we do all of the ethical submissions and approval, we get all of the contracts drafted and sorted with the hospitals, we get standardized clinical kits sent to all of the different sites that may be running in parallel. These contain all of the, tourniquets, the vacutainers, the needles, anonymized, labeled, barcoded tubes. And then we have the sample shipped to Milan, to our partner BioRep, where it's biobanked, and then aliquots are sent to us in Dublin for analysis. And so for this small study, it comprised of 16 healthy controls, 10 individuals with PMP22 duplication, so CMT1A, 3 patients with MFN2 duplication, CMT2A, and then 5 with PMP22 deletion, so HMPP. And this was done with Dr. Sinead Murphy, University Tallaght Hospital. So what we first did is, in this very small cohort of patients, see can we, replicate what we see in other studies. And we did see a degree of an increase in neurofilament light. The low N numbers and the variability, didn't reach significance in CMT1A. And then when we compare this to the CMTS clinical rating measure, you get a bit of a kind of confusing graph here. It doesn't really show anything. It's very low N numbers. And then when we looked at acetylated alpha-tubulin, we did see a degree of a reduction in acetylated alpha-tubulin in this small cohort of patients, with potentially some degree of correlation there. They seem to be moving in the right direction across the 3 patient subtypes. But it was only the HNPP that reached statistical significance. But we can't really draw any conclusions from these data with such a small sample size. So then we moved to a follow-up study, and we increased this from Ireland to Italy as well. And so we again worked with Dr. Sinead Murphy in Tallaght, and we recruited a further 15 controls, and then 6 more CMT1A patients that were different to the CMT1A patients in the first study. And then we worked with Dr. Stefano Previtali in Sarafella Hospital in Milan, who also recruited 16 healthy controls and 39 CMT1A patients. So a total of 31 healthy controls and 45 CMT1A. Here we managed to see a much more robust increase in neurofilament light chain. There is still a degree of variability. And when we look at correlations with age, we did see a degree of correlation with age in the control population, not so much in the CMT1A population. When we then compare these to the 2 clinical outcome measures that we had for these patients, which was CMTS and ONLS, we didn't see any statistically significant correlation with the severity of their CMT1A with the levels of NFL, which has been reported in the past. Then when we looked at the levels of acetylated alpha-tubulin, we did see a nice significant decrease in the levels of acetylated alpha-tubulin. Important to note here that the technique we're using for measuring this is multiplexed infrared Western blotting, where we're measuring the levels of acetylated alpha-tubulin normalized to levels of total alpha-tubulin. This is great for a basic science principle, but if we want to move this forward, we need to consider how can we generate this into a standardized methodology and assay, which we're currently working on. When we looked at this levels compared to age, we saw absolutely no correlation here, and this levels seem to be fairly consistent across the age range that we looked at. And then when we compared this to, again, the 2 same functional outcome or the clinical outcome scores, we did see a statistically significant correlation with the levels of acetylated alpha-tubulin and increased severity in these patients. This was only a cross-sectional study, and there was no longitudinal analysis here. So obviously the next steps would be to see whether we can follow up with the same patients again at later time points and see whether this holds up over a longer period of time. So I kind of whizzed through that, but in summary, in these two studies that we ran, we saw, as we expected, NFL is significantly increased in the plasma of patients, but doesn't necessarily correlate with any of the clinical outcome scores that were provided by the clinicians. We saw a highly significant reduction in acetylated α-Tubulin in the plasma of these patients, and this reduction in acetylated α-Tubulin shows strong inverse correlation with increases in the severity of these patients. As I said, next steps would be to, make this into a more robust and validated assay, and also to continue looking into a more longitudinal cohort of patients. And I will leave you this quote here, which we live by at Ulysses Neuroscience, and thank you very much for listening.
We have time for one question. Stephen, sure.
So I missed the number of controls that you have used. So did you use young people, elder people? Because we all get older, and then probably tubulin acetylation will change over time.
So in the second follow-up study, we had 31 controls, 45 CMT1A, and if I remember correctly, the age range was all the way from 21 up to 73, I think was the age range we had. We do have other graphs, and we're preparing a manuscript at the moment where we do dig into correlations with age. Just for the sake of time today, I wanted to just show the key results. But yeah, there is, for acetylated α-Tubulin, we see no correlation with age at all in the controls or in the CMT1A patients. In the CMT1A patients, we also see no correlation with age in the CMT1A patients for NFL. But then for the controls, we do see a correlation with age in the NFL.
Maybe a last question from Steve Scher.
It was
Steve?
Steve?
Sorry, I was looking there.
So there are some details that you know that you didn't tell us. This alpha-tubulin is only axonal that's acetylated that you're measuring. Is that true?
So we're taking blood samples here. So we, proving that it's coming from the brain is one aspect is, whether it's peripheral or whether it's central, because we've looked at this in other disorders as well, and looked in CSF and plasma. In terms of whether it's axonal,
or at least neuronal / axonal, it's coming from, there's no other cell type that expresses this particular tubulin gene.
Exactly, yeah. So this is neuronal, we believe, and one way we've looked to confirm this is looking at the levels of another modification of α-Tubulin, which is the delta 2 modification. So it's the removal of the terminal 2 residues of the protein, which only occurs in neurons.
And in animal models that are given HDAC6 inhibitors, is that treatment responsive biomarker?
We haven't personally done that. We actually don't have any CMT animal models that we're working with at the moment. So I don't know about that. It'd be interesting to see.